During vigorous exercise, the output of the heart is increased and local blood flow patterns change. The result is an increased blood supply to the muscles at the temporary expense of other organs. These changes are integrated and blood pressure remains relatively constant. High blood pressure can cause vascular damage, particularly in the kidneys and brain. We are now going to consider some of the mechanisms involved in cardiovascular control, both at the heart and in the peripheral circulation. First, let's examine the local regulation of blood flow to the tissues of the forearm. At the extremities of the blood system, the arterioles break up into countless microscopic capillaries. Blood flow through these networks can be continually altered to meet metabolic demands, and this is evident if the blood supply is temporarily interrupted. We can measure the blood flow to the forearm by the technique of plethysmography, where cuffs are applied to the wrist and upper arm. These can be inflated through a range of pressures to occlude venous or arterial blood flow. A strain gauge is applied to the limb, and this will measure any changes in the dimension of the forearm that occur. The hand is first excluded from the circulation by inflating the cuff on the wrist to above systolic pressure. The cuff on the upper arm is then inflated to a lower pressure for a period of about 10 seconds to occlude the veins. When this happens, all the blood flowing into the forearm is trapped and the limb progressively swells. The rate of swelling is recorded on a chart, the slope of the line being proportional to the blood flow. The pressure on the upper cuff is then released, allowing the blood to flow out of the limb. This operation is repeated several times to determine the average resting forearm blood flow. We now inflate the upper cuff to above systolic pressure to cut off the blood supply to the whole forearm. After 1 minute, we deflate the upper cuff and measure blood flow, again using the same technique. It can be seen that there is an immediate and large increase in flow, followed quite quickly by a return to the control level. Here is the complete recording, showing the normal resting flow, the period of occlusion, and the increased blood flow that occurs when the upper cuff is relaxed. If the hand is exercised during the occlusion period, an even greater increase in blood flow is observed when the cuff is released. How are such changes explained? Nutrients and metabolites are exchanged across the capillary. When the blood flow is interrupted, metabolites like ADP and lactate may accumulate, and the partial pressures of oxygen and carbon dioxide may change. Such factors can induce relaxation of precapillary sphincters and arteriolar smooth muscle. This process is called autoregulation. When the cuff is released, the increased blood flow that results quickly removes the accumulated vasodilatory metabolites, and resting equilibrium is regained. Blood flow is also regulated by extrinsic controls, which may be hormonal or nervous. If a cannula is inserted into the brachial artery, it is possible to investigate the effects of neurotransmitters or hormones on blood flow. The cannula is connected to a syringe driver, through which we can infuse either a saline solution or noradrenaline. We first measure resting forearm blood flow. The arterial line is then transferred from the saline to the second syringe containing noradrenaline. Here are the results. There's a considerable reduction in blood flow when noradrenaline is added. Flow returns to normal when the saline infusion is restored. This preparation has been treated to convert noradrenaline into a fluorescent substance, and this delicate tracery indicates that arterioles are richly innervated with adrenergic nerve fibers. Increased activity in these nerves cause arteriolar smooth muscle to contract. Here's an arteriole with its sympathetic innervation, and now we can explain why noradrenaline reduces peripheral blood flow. There is always some degree of sympathetic smooth muscle tone in arterioles. By increasing sympathetic activity or perfusing noradrenaline, further constriction can be produced. By reducing the rate of sympathetic discharge to below the basal level, we get vasodilation. This enables the nervous system to exert a very fine control over the microcirculation. Individual arterioles, like this one on the right, can be shut down, while others remain open. The second site of cardiovascular control is the heart. Cardiac performance can be investigated by linking the left side of a heart to an artificial circulatory system, where it's possible to control the resistance into which the heart pumps and the pressure of the blood returning to the atria. Now look at the principal elements of this isolated heart preparation. Oxygenated blood flows from the reservoir into the supply container. This is connected to the heart and to an overflow system. The overflow container can be raised or lowered to adjust the height of the column of blood in the supply container, which in turn varies the filling pressure of the left atrium. Blood pumped from the heart then passes through the model of the arterial circulation with its adjustable resistance. This resistance consists of numerous conduits, and by moving a slide across them, a greater or lesser number is occluded, varying the resistance to flow. The air chamber simulates the natural elasticity of the arteries. This part of the model reproduces the properties of the aorta. The measurements taken from this system include aortic pressure and flow, left ventricular pressure, and ECG. The cardiac output and stroke volume are computed from these data. The information obtained from these experiments is displayed on magic tracings, and the measuring equipment allows us to record the appropriate data simultaneously. The first trace is a time scale, each pulse equaling 1 second. Below this is a recording of the ECG. The third trace shows the pressure in the aorta, the peaks registering systolic and the troughs diastolic pressure. The fourth trace represents left ventricular pressure, with the height of the trace indicating peak systolic pressure, and derived from this is the dp/dt trace. The height of this trace gives the maximum rate at which the pressure rises, and is an index of the strength with which the ventricle is contracting. The next trace represents aortic flow, and from this, cardiac output and stroke volume are computed. In our first experiments, we shall pace the heart at a constant rate using a pacemaker. This means that any change in cardiac output reflects changes only in stroke volume. The final trace shows this constant heart rate. We first set heart rate, peripheral resistance, and filling pressure to specific values. And we call these our control settings. Under these conditions, all measurements reach a steady value. Now let's study the effect of two interventions. We begin by increasing the peripheral resistance while maintaining the same filling pressure and heart rate. This increased resistance means that it's harder for the heart to force blood through the system, and it can be seen that this leads to a rise in the aortic and left ventricular pressures. As a result, both cardiac output and stroke volume fall because the heart is now pumping against an increased resistance. This model shows what happens in the heart when there are changes in the peripheral resistance indicated by the arrow. If the filling pressure and resistance are constant, the end diastolic and end systolic volumes will also be constant. If the peripheral resistance goes up, the heart can eject less blood, and so there's an increase in the end systolic volume. In other words, there's a less complete emptying of the ventricle. A reduction in the peripheral resistance would lead to an increased cardiac output and a reduced end systolic volume. Let's examine now the effect of another intervention. We increase the filling pressure by raising the overflow container, and the result is that cardiac output goes up. Because the heart is paced at a constant rate, this increase is due solely to an increase in stroke volume. As you would expect, aortic and left ventricular pressures also increase as a consequence of the greater amount of blood being ejected against the same resistance. This increase in cardiac output occurs because raising the filling pressure causes the ventricles to stretch, so increasing end diastolic volume. This is an intrinsic control mechanism deriving from an inherent property of muscle tissue, which in the heart is known as the Frank-Starling mechanism. Let's review the sequence of events again using the model of the ventricle. When the filling pressure is raised, end diastolic volume increases. This in turn increases the tension on the ventricular walls, and a more forceful contraction results. This represents Starling's law of the heart. Note that there is no change in ventricular end systolic volume. During strenuous activity, much more profound adjustments have to be made in cardiac performance. Cardiac output may be increased up to 6 times to maintain the supply of oxygen to the active muscles. This change results from extrinsic controls via the autonomic nervous system. 2 types of autonomic nerves control heart rate: sympathetic accelerator nerves emerging from the spinal cord, and parasympathetic fibers arising from the medulla passing to the heart via the vagus nerve. Acetylcholine released from the vagal nerve decreases the discharge rate of the pacemaker cells in the sinoatrial node, so heart rate decreases. Sympathetic stimulation causes noradrenaline to be released, and this increases the discharge rate. The intrinsic discharge rate of the SA node is about 80 a minute, but normally the heart rate is about 70 beats a minute, indicating a predominance of vagal tone. Heart rate can be increased, therefore, either by a reduction in vagal tone or by an increase in sympathetic tone. Noradrenaline also controls the contractility of the ventricles, and this has an important bearing on cardiac performance. In times of stress, another sympathetic mediator, the hormone adrenaline, is released into the bloodstream from the adrenal medulla. This acts on both the SA node and the myocardium, reinforcing the effect of locally released noradrenaline from the sympathetic nerve terminals. We can reproduce the effect of autonomic stimulation and of hormones in our isolated heart preparation. The pacer used earlier is first switched off, and a solution of noradrenaline is injected into the left atrium. As you would expect, there's an increase in heart rate and cardiac output. With noradrenaline, not only is there an increase in rate, but the heart also beats more forcefully. When we look at the aortic and left ventricular pressure traces, we find that pressures go up because the higher blood flow is being pumped through the same peripheral resistance. However, there is now also an increase in the dP/dt, that is the maximum rate of rise of left ventricular pressure, which is a measure of the strength of contraction. If you inject adrenaline, the effects are similar. With each catecholamine, not only is the contraction more powerful, it occurs more rapidly as well. Relaxation too is faster. This helps to increase the time available for ventricular filling, while as a consequence of the more forceful contraction, there's a reduction in end systolic volume. The result is an increase in stroke volume, and this, combined with a higher heart rate, raises cardiac output. So noradrenaline improves heart performance independently of the Starling mechanism. There is an increased contraction with no change in end diastolic volume or initial muscle fiber length. In the brain, capillaries are particularly sensitive to changes in blood pressure. Cardiovascular performance is continuously monitored by pressure sensors in different parts of the blood system, one of which is in the carotid artery. Information from these baroreceptors is relayed to the brain, which regulates the autonomic nervous system so that blood pressure is kept relatively constant. Now let's look at the properties of the carotid baroreceptor. At the origin of the internal carotid artery supplying the head and brain, there's a specialized elastic dilatation called the carotid sinus, which contains a large number of branching nerve endings in the tissue. The wall of the carotid sinus is much more distensible than the adjacent artery and stretches in response to changes in arterial pressure. The nerve endings are sensitive to stretch and send out bursts of impulses. The frequency of discharge is linearly related to changes in stretch over the normal range of pressures. The vagus nerve carries information from the baroreceptors in the aorta, whereas the glossopharyngeal nerve carries the afferent axons that come from the carotid baroreceptors via the carotid sinus nerve. In order to isolate the carotid baroreceptors for study, the vagus nerve is cut, and cannulae are inserted to make a loop with a sidearm. Two clamps, C1 and C2, are incorporated into the circuit. These are used to perfuse the sinus area, either with the animal's own blood, by closing C2 and opening C1, so that blood goes around the loop, or by closing C1 and opening C2, we can perfuse it with saline through a pressure generator connected to the sidearm. A further cannula with a pressure transducer is inserted into the area of the carotid sinus through the lingual artery. This is used to measure the pressure in the carotid sinus, P2. A pressure transducer in the femoral artery records the arterial blood pressure, P1. Heart rate is displayed on a digital counter, which records the rate per minute. Pressure shown on this gauge indicates either arterial pressure, P1, or the pressure in the carotid sinus, P2. In order to investigate how the baroreceptor influences the cardiovascular system, it is necessary to monitor several parameters. We will first measure arterial blood pressure, carotid sinus pressure on the middle trace, and finally heart rate. Initially, clamp 1 is opened so that the sinus is perfused with the animal's own blood. The pressure in the carotid sinus, P2, is therefore normal. If the blood supply to the carotid sinus is now cut off by closing clamp C1, the carotid sinus pressure falls to a low level, thus stimulating the baroreceptors. Heart rate immediately increases dramatically as the autonomic nervous system senses and tries to compensate for the fall in carotid pressure. Consequently, arterial pressure, P1, rises to a very high level, mainly because of peripheral vasoconstriction. When blood is allowed to flow into the carotid sinus again by loosening the clamp, P2 rises to the same level as P1, as both are now measuring arterial pressure. Almost immediately, the baroreceptors exert their influence to reduce the high level of pressure to within the control range. There is also intense bradycardia, so that the heart rate actually falls below control level before stabilizing again. Here are the results of the experiment. Before the blood supply to the carotid sinus was cut off, both P1 and P2 were registering arterial blood pressure. P1 follows a little behind P2, since the pulse wave reaches the femoral artery slightly later than the carotid sinus. When the clamp is applied, the pressure in the carotid sinus falls, and the heart rate and arterial pressure rise, mainly because of changes in sympathetic activity. When the clamp is released, P2 immediately rises to the same as P1, since both are now registering arterial pressure. Both pressures will now fall as the autonomic balance is adjusted to restore blood pressure to the control level by restoration of normal vagal tone and sympathetic tone in the peripheral circulation. So in this trace, we see evidence that the baroreceptors can perceive changes in carotid sinus pressure and convey this information to the vasomotor centers of the brain, which can modify the efferent impulses to the heart and the sympathetic tone in the peripheral circulation. Changes in blood pressure alter the dimensions of the carotid sinus, which you can measure using ultrasound crystals placed on each side of the sinus. One crystal sends out a sound pulse, and the time taken for this pulse to reach the second crystal is related to the dimensions of the sinus. The activity of the nerve axons leaving the sinus can be monitored at the same time by splitting the nerve sheath and placing a single nerve axon onto a pair of platinum electrodes. The output of the nerve is only about 50 millivolts, and the signal has to be amplified several thousand times. Clamp C2 is now opened so that the sinus can be perfused from an artificial pressure generator which produces a pulsatile pressure similar to that delivered through the animal's own cardiovascular system. The recordings show the pressure and the changes in dimension in the carotid sinus. The bottom trace displays the individual action potentials traveling down the fiber, seen as separate spikes and heard as clicks. At low pressure, the nerve output is sporadic, and there is no clear association with pressure or dimension changes in the carotid sinus. At a pressure in the middle of the physiological range, the nerve fibers fire in short bursts, registering the pressure and dimension changes in each cycle. As the pressure rises further, the length and intensity of the bursts of firing increasing. Let's look at both the afferent and efferent pathways involved. The information from the baroreceptors goes to the medulla. This region of the brain initiates changes in the activity of the autonomic nervous system via the vagus nerve, shown in blue, and sympathetic nerves, shown in red, which pass to the heart and to the arterioles. When arterial pressure falls, the walls of the carotid sinus distend less. Consequently, the discharge frequency of the baroreceptor nerves is reduced. The result is a reduction in the output of the vagus nerve and an increase in the activity of the sympathetic nerves, resulting in an increase in cardiac output and vascular tone. The net effect is to restore blood pressure to its previous levels. A rise in arterial pressure produces the opposite effect. The carotid sinus nerves increase their frequency of discharge. Vagal tone increases, and there's a corresponding decrease in sympathetic output at the heart and at the arterioles. The combination of a reduced cardiac output and peripheral vasodilation restores the blood pressure to normal. This system normally ensures that blood pressure does not reach dangerously high levels, even during the most strenuous activity. Although the main elements of the control system are known, many of the fine details are still not fully understood. However, we do know that the fine control is so wonderfully precise that the circulatory system is able to support even these record-breaking efforts.
In the intact heart, the bulk of the myocardium is composed of contractile myocardial cells. That is, cells which are capable of contraction, but do not possess intrinsic rhythmicity. Pacemaker cells, that is, cells which possess intrinsic rhythmicity, are associated with a conduction system embedded in the myocardium, which is responsible for the coordinated activation and contraction of the various parts of the heart. Within the myocardium, electrical impulses can be transmitted from one cell to the next in any direction. This process is termed conduction. However, once an impulse starts traveling in one direction, it cannot travel in the opposite direction because the cells which it leaves behind are always in their absolute refractory period. This property of the myocardium ensures coordinated contraction in the normal heart. Impulses originating in the sinoatrial node spread through conduction pathways in the right atrium. These impulses are then distributed to all parts of the right and left atria, causing them to contract. The presence of two fibrous rings surrounding the atrioventricular valves provide an effective separation of the atrial myocardium from the ventricular myocardium, except at a single point. This is the atrioventricular node, which lies at the base of the interatrial septum and runs into the fibrous part of the interventricular septum. The atrioventricular node and the main bundle of His are known as the atrioventricular junction. The impulse is now conducted through the two branches of the bundle of His, which lie just beneath the endocardium of the right and left ventricles respectively, to reach the apex of the heart. Here, each bundle turns upwards and passes towards the base of the heart, thus forming a right and left bundle branch. Here we see the conducting and activating system in action. The sinoatrial node discharges. Activating impulses reach the atria, and they start to contract, boosting ventricular diastolic filling. Now impulses reach the atrioventricular node, where their passage is delayed. By the time the impulse has reached the bundle of His, atrial systole is completed. Rapid conduction of the impulse through the branches of the bundle of His activates the Purkinje system, and a coordinated activation and contraction of the ventricles takes place. Because of the organization of the ventricular conduction system, different parts of the ventricular myocardium are activated successively. As a result, a peristaltic-like wave of contraction passes over the ventricles, ensuring the most effective ejection of the blood within them into the pulmonary and systemic circuits. This sequence is repeated at the rate imposed by the sinoatrial node.
This 45-year-old man was found by his doctor to have a blood pressure of 150/100 at a routine medical examination. An elevated blood pressure of this order is not infrequently due to the anxiety many people experience when they undergo a medical examination. In our patient, the blood pressure did not return to normal under basal conditions, and further readings taken over the next 3 weeks showed that he had a persistently elevated blood pressure of about 150/100. It could be concluded, therefore, that he was suffering from hypertension. The history, physical examination, and routine investigations failed to disclose any primary cause for the hypertension. Furthermore, they did not provide any evidence of serious secondary damage due to the hypertension itself. The doctor therefore felt justified in making a diagnosis of uncomplicated essential hypertension. Because some vascular systems withstand the effects of high blood pressure better than others, it is not possible to predict the extent of damage that will result from an elevated blood pressure in any particular patient. Nevertheless, when we look at insurance company actuarial statistics regarding hypertension and involving many millions of people, some important facts emerge. At the age of 55 years, a blood pressure of 150/100, if untreated, is associated with a reduction in life expectation of 6 years. At the age of 45 years, as in our patient, it is associated with a reduction in life expectation of 11.5 years. And at the age of 35 years, a reading of 150/100 is associated with a reduction in life expectation of 16.5 years. Lowering the blood pressure in such patients has been shown to have a significant effect in increasing life expectancy. There was therefore a clear indication for antihypertensive therapy in our patient with a blood pressure of 150/100. The doctor advised the patient to reduce weight and prescribed a thiazide diuretic together with a potassium supplement. This treatment resulted in a fall in the patient's blood pressure to 130/90. These are cardiac output, peripheral resistance, and blood volume. All drugs with an antihypertensive action exert their effects by acting on one or more of these parameters. Let us consider each of these in turn and see how they are affected by antihypertensive agents. The cardiac output depends partly on the heart rate and partly on the stroke volume of the left ventricle. These two factors are influenced by sympathetic impulses reaching the heart via the autonomic nervous system. An increase in sympathetic activity produces an increase in heart rate and in stroke volume, increasing cardiac output, thus tending to raise blood pressure. Beta-adrenergic blocking agents act partially by selectively counteracting the effects of sympathetic activity on the heart, reducing heart rate and stroke volume, with a consequent fall in cardiac output and thus blood pressure. Peripheral resistance depends on vasoconstrictor impulses reaching the smaller arterioles via sympathetic fibers. An increase in sympathetic activity causes an increase in arteriolar tone, which produces an increase in peripheral resistance and thus an increase in blood pressure. Most drugs used to reduce peripheral resistance act by lowering the effective concentration of noradrenaline at the synapse between the peripheral nerve terminal and the smooth muscle fibers of the arteriolar wall. In these circumstances, the arterioles relax, reducing the peripheral resistance and, consequently, the blood pressure. As we shall see later, blood volume depends principally on the total body content of sodium. An increase in body sodium produces an increase in blood volume and thus an increase in blood pressure. Thiazide diuretics act mainly by decreasing the sodium content of the body, reducing blood volume and thus reducing blood pressure.
The 20s saw the first stage of the triphasic shift in diuretic antihypertensive therapy. Salt restriction. It meant a monotonous and unpalatable diet, but it was moderately effective in some patients. The second generation is well exemplified by the thiazide diuretics. They are effective and widely used. But thiazide therapy on its own can result in complications. They can elevate blood urate levels. This may be a minor risk factor in the etiology of coronary heart disease, and may, in the gout-prone individual, precipitate an acute gout attack or cause renal damage. Thiazides reduce sodium reabsorption in the distal tubule, with a consequent increase in sodium-potassium exchange and urinary potassium loss. The diuresis-induced hypovolemia activates the renin-angiotensin-aldosterone sequence. This increase in aldosterone causes both sodium retention, which buffers the antihypertensive effect, and a further loss of potassium. This could result in hypokalemia, which may be associated with certain side effects. In severe cases, these include renal tubular damage, cardiac arrhythmias, particularly in digitalized patients, and paralytic ileus. Other possible side effects include constipation, muscle weakness, and anorexia. The complications which could arise from thiazide therapy emphasize the need to monitor serum potassium in patients during the first 2 months of therapy, and then annually. There are various methods commonly used to correct the potassium loss associated with thiazide therapy. The thiazide can be intermittently withdrawn, the so-called drug holiday. An alternative is to co-prescribe a potassium supplement, preferably as potassium chloride, thus avoiding hypochloremic alkalosis. Unfortunately, potassium chloride is unpalatable and may cause gastric irritation. Enteric capsules have proved liable to disintegrate at one site in the small bowel, causing ulceration, or may not dissolve at all. Slow-release preparations are better, although there are isolated reports of lodgement of the capsule in the esophagus, with subsequent ulceration and stricture. Tablets containing a combination of a diuretic and potassium chloride generally contain insufficient potassium, And if potassium is given as a supplement, as many as 6 tablets may be required daily. Moreover, the potassium may be better retained if not given on the same day or at the same time as the diuretic. This necessitates complex dosing schedules and could lead to poor patient compliance.
There are 2 main problem areas in the treatment of hypertensive patients. The first concerns the difficulty of individual patient monitoring, and the second, the problem of non-compliance. Patient monitoring is necessary for a number of reasons. Apart from taking the blood pressure, it is important to look for such developments as adverse drug reactions, potential drug interactions, or electrolyte imbalance. The second difficulty of patient compliance is due to 2 main factors. Hypertension is generally an asymptomatic condition, and the range of treatments available for this condition may have unpleasant side effects. This, taken with an often complex dosage schedule, can lead to patient non-compliance as high as 50%.
2 whenever required, but don't take them more than 4 times a day.
The ideal antihypertensive should normalize the blood pressure with a minimum of short and long-term side effects. It should ideally be available in a once-daily formulation. We have seen that the thiazides, although well developed as first-line drugs, are by no means perfect. They have a range of side effects, including those of potassium imbalance, which may complicate patient management.